Good day, ladies and gentlemen. You are welcome to BCH 408, Topic 2, titled Metabolism and Regulation of Heme. In our last class, we had a first lecture. Can you remember the topic? Okay, good. Today, we are going to look at metabolism and regulation of heme, and I promised you that whatever matters are arising from the first video class, which I meet physically to attend to them. When that time is due, I will pass the information across to you. But today, our learning objective are ninefold. At the end of this lecture, you are expected to be able to define heme, his chemical structure and biological significance, identify the major components in the body of hemoproteins in the body, describe the site and locations of heme synthesis, outline the steps and key enzymes involved in heme biosynthesis, explain the mechanism regulating heme biosynthesis. You should be able also to describe the pathway of heme biodegradation, explain the transport, conjugation, as well as excretion of bilirubin, discuss disorders associated with abnormalities in heme metabolism, and finally, you should be able to evaluate the significance or discuss the significance of heme metabolism. Now, without wasting time, let us go straight to defining heme, its structure and significance. Heme simply is an iron-containing porphyrin compound that consists of protoporphyrin 9 ring coordinated with a central ferrous ion. Now, the definition is simple. Now, this is what I refer to. This is porphyrin 9, protoporphyrin 9, and this is the ion at the middle. It is attached to the nitrogen atom of the protoporphyrin 9 with the protruding molecules here. You can see M, you can see V, you can see P. Now, P stands for propionyl, M stands for methyl, and V stands for vinyl. Now, these are the tails. We refer to them as tails of the heme molecule, that is, housing the ion atom. Now, before we go further, let us take a look at hemoproteins. What are they? They are simply heme combined with proteins. These proteins contain heme as their prosthetic group. In these proteins, the heme molecule, which is protoporphyrin 9 with the ion, is tightly bound to the protein structure. It is this binding that enables the protein to perform its biological functions. That is, the binding between protoporphyrin 9 with ion. With the absence of ion at the central core of the molecule, that protein cannot be functional, because The ion atom in the center of the heme group is the active center. Now, we have talked about the hemoproteins being functional biological molecules. What is their significance? One, they help in oxygen transport, oxygen storage, electron transport, enzymatic analysis, catalysis, I beg your pardon, regulation of cellular metabolism, and so on and so forth, with some minor activity and classified with them. Of course, they also have some clinical importance. Now, let's look at the major types of these hemoproteins and their functions. In oxygen transport, hemoproteins like hemoglobin in red blood cells and myoglobin in tissues or in muscle cells have biological role to play. Also, we also have the cytochromes playing the role of electron transport hemoproteins. We have catalase and peroxidases playing the function in enzymatic catalysis, and cytochrome P450 in detoxification reactions. Now we shall take these functions one after the other, that is, one by one. Now let's start with oxygen transport hemoproteins. As mentioned earlier, they are found in hemoglobin and myoglobin. In hemoglobin, they play the role of transport of oxygen from the lungs to the tissues, as well as carrying or transporting carbon dioxide from tissues to lungs. In myoglobin, that is in muscle tissue, they store oxygen for muscular metabolism, where they help supply oxygen during muscular contraction or strenuous muscular exercises. The cytochrome, on the other hand, they are located in the mitochondrial electron transport chain and are involved in oxidative phosphorylation. The types include cytochrome A, cytochrome B, cytochrome C, and cytochrome oxidase. Now, cytochrome P450 is another class that are involved in detoxification reactions. They are predominant in the liver and endoplasmic reticulum, where they are involved in drug metabolism. They oxidize drugs, toxins, steroids to make them easier for the body to eliminate. Now, we have talked about heme, we have talked about hemoproteins, we have talked about their functions and their significance. Now, let's look at the heme itself. Where are the locations of heme sequences? We can really mention two, that is the bone marrow and the liver. Bone marrow for hemoglobin production, liver for cytochrome and metabolic enzymes, and the parts in which they occur are the mitochondria and partly in the cytosol. The first and last step of the heme synthesis take place in mitochondria, while the other intermediate step occur in the cytoplasm. Now, much said about the preambles to heme biosynthesis. Now, let's look at biosynthesis of heme. Now, the biosynthesis of heme requires substrates. The substrates are glycine, succinyl-CoA, iron, and vitamin B6 or pyridoxal phosphate. Heme synthesis involve 8 enzymatic reactions, wherein the pathway begins with glycine and succinyl-CoA. The first step is delta aminolevulinic acid, which is called in short ALA. And the final step involves insertion of iron into protoporphyrin 9. Now, this is an overview of heme synthesis. As I mentioned earlier, heme synthesis start from glycine and succinyl-CoA through a series of 8 enzymatic reactions. The pathway occurs partly in the mitochondria and partly in the cytoplasm. The first step occurs in the mitochondria. The intermediate step occurs in the cytoplasm. In the first step, this pathway eventually produces heme. Now, let's look at the biosynthetic pathway. The first reaction is the formation of ALA, that is delta aminolevulinic acid. That is the substrate glycine combined with succinyl-CoA, and the enzyme is alpha aminolevulinic acid synthase, that is DALAS in short. The second step involves formation of porphobilinogen in the cytoplasm. Now, that reaction, formation of porphobilinogen, is catalyzed by alpha aminolevulinic acid dehydratase. That is, in short, DALAD. ALAD. Now, the third step, as you can see, involves the formation of hydroxymethyl bilane, and the enzyme responsible for that catalysis is porphobilinogen deaminase. The fourth step is formation of uroporphyrinogen 3. Uroporphyrinogen 3 is catalyzed by the enzyme uroporphyrinogen 3 synthase, that is UROS. UROS. Fifth step is the formation of coproporphyrinogen 3, and the enzyme responsible is uroporphyrinogen decarboxylase. Now, the sixth step is the formation of protoporphyrinogen 9, and the enzyme is coprotoporphyrinogen oxidase. Now, the seventh step is the formation of protoporphyrin 9. Protoporphyrin 9, and that the enzyme responsible for that is protoporphyrinogen oxidase. While the last step, which is the formation, which leads to the formation of heme, is catalyzed by the enzyme ferrochelatase. You can see now, having talked about the synthesis, let's talk about the breakdown or the catabolism of heme. This often occurs when the heme or blood becomes old and damaged. They have to be removed from circulation by macrophages. Now, the cells that are involved is the spleen, the liver, and the bone marrow. During this process, heme is released from erythrocytes. Now, the heme that is released from the erythrocytes or the red blood cell is separated into two parts, which is heme and globin. Now, the globin is further broken down to amino acids, which are reused by the body, while heme undergoes enzymatic degradation. Now, what pattern or reaction does heme degradation or is heme degradation involved in? Here is it. The porphyrin ring of heme is cleaved. The iron atom is removed. Carbon dioxide is produced. And the product you get is biliverdin, iron 3, carbon monoxide. Those are the three products that are released. The iron is recycled and transported by transferrin to the bone marrow for reuse. Now, here in the white part is a pathway for your convenience to further your understanding of the process. You can see the heme there. You can see heme oxygenase that breaks down, that catalyzes the splitting of heme to biliverdin. Now, from biliverdin reductase using NADPH will now oxidize biliverdin to bilirubin. Before we go further, let's ask ourselves the characteristics of bilirubin. You must note that bilirubin has 4 characteristics. 1, it is yellow. It is a yellow pigment. 2, it is water insoluble. 3, it is transported in the blood, bound to albumin. And 4thly, the complex travels to the liver for further metabolism. Further metabolism in this case means a reaction, or simply put, phase 2 conjugation reaction, whereby the liver metabolizes this molecule and make it more water soluble for easy excretion from the body. Now, what does the conjugation reaction looks like? The conjugation reaction is catalyzed by UDP glucuronyl transferase. That enzyme attaches glucuronic acid to bilirubin. It attaches 2 molecules of glucuronic acid to bilirubin. And when that is done, bilirubin becomes water soluble and secreted into the bile. From where it is excreted by the kidney. Now, that said, we've talked about metabolism, degradation, and significance of this process. Is it possible to have disorders in heme metabolism? Yes, it is very possible. This can occur as a result of enzymatic problem or enzyme problem the cell faces during synthesis of heme or degradation or breakdown of heme. And these defects or disorders are referred to as porphyrias. They are referred to as porphyrias. Porphyria or porphyrias can be divided or are divided into acute or non-acute forms. In acute porphyrias, you have acute intermittent porphyria, variegated porphyria, hereditary coprotoporphyria, and DOS porphyria. In non-acute porphyria, however, you have porphyria cutanea tarda, hepatoerythropoietic porphyria, erythropoietic protoporphyria, protoporphyria, and X-linked protoporphyria. We also have congenital erythropoietic porphyria. Now, let's look at causes of heme disorders. Heme disorders may arise when there is a problem with making, using, or breaking down heme, usually due to enzyme or gene defects that disrupt the heme synthesis pathway. These problems could lead to deficient heme production and buildup of toxic intermediates that damage tissues. Some of these causes could be attributed to 1. Inherited enzyme defects in heme synthesis. 2. Accumulation of porphyrin intermediates. 3. Defects in heme transport or handling in erythroid cells. 4. Gain of function mutations increasing heme precursors. In this case, we talk of accumulation. The accumulation arises because enzymes that are supposed to convert substrates to products are either lacking or deficient. But in item 4 here, we are talking of gain of function mutations, increasing heme precursors. Under gain of function mutations, in this case, the enzyme is not deficient, but it is rather excessively produced, so that more products will be produced, production of more products will be enhanced. 5. Nutritional and acquired factors affecting heme enzymes. That is, when you don't have the corresponding or requisite factors that are necessary to produce the necessary enzymes that are required to produce these enzymes that are involved in the heme synthetic or biosynthetic pathway. Drugs, toxins, and hormones can also can cause this disorder. That is, shortage or deficiency of hormones, use of drugs, and toxins can also be a factor. 7 is liver disease and iron overload. Liver disease is a major factor because when the liver is diseased, then a lot of metabolic problem will ensue. Finally, number 8, we have associated hemoglobin and red cell disorders. When the red cell is disordered, and in fact, if you have a disordered red blood cell, of course, the amount of hemoglobin you are likely to have in the cell will be disrupted. And that could be a very serious factor. Now, in all, what is the significance of heme metabolism? Heme metabolism is crucial because it enables oxygen transport and energy production, supports detoxification and signaling, and safely handles the potential toxic heme molecules to maintain overall tissue and organ health. Now, key physiological rules involved oxygen transport and storage, cellular energy production, detoxification and metabolism, signaling and gene regulation, finally, safe disposal and recycling, as we saw in the case of the globin being recycled and the iron also being recycled during heme biosynthesis and degradation. Now, much has been said. It is now time for you to appraise yourself, to know your level of comprehension on what I have just delivered to you. So for that, I have given you a set of 5 study questions you can appraise yourself with. Take your time to go through them. By going through them, you'll be able to appraise yourself whether or not you have adequately comprehended what have been delivered today. My dear students, I want to thank you very much for your time. This is the end of lecture 2. We'll look forward to lecture 3 soonest. Thank you and God bless.